Hello everyone. Today we are going to discuss hallmarks of cancer. So what is the meaning of hallmarks of cancer? It's basically referring the characteristics that distinguish the cancer cells from the normal cells. एक नॉर्मल सेल और कैंसर सेल में क्या डिफरेंस है, कैसे हम अंतर लगा सकते हैं कि दोनों में क्या ऐसा चीज़ें हैं देख करके कि वो कैंसर सेल है और एक नॉर्मल सेल है. ठीक है. सबसे पहले इसको साइंटिस्ट रॉबर्ट विनबर्ग और डगलस हैनन ने 2000 में दिया था और इसको अपडेट किया गया 2011 में. इसमें हम मुख्यतः कुछ पॉइंट्स पे डिस्कस करेंगे. वो सारे पॉइंट्स क्या क्या हैं. जो स्क्रीन पे दिख रहे ये सारे पॉइंट्स हैं. इंक्रीज्ड एक्शन ऑफ पॉजिटिव ग्रोथ रेगुलेटर्स. दूसरा, लॉस ऑफ फंक्शन ऑफ निगेटिव ग्रोथ रेगुलेटर्स. थर्ड इज अल्टर्ड सेलुलर मेटाबॉलिज्म. फोर्थ इज लॉस ऑफ नॉर्मल एपोप्टोसिस पाथवेज. फिफ्थ वन इज लॉस ऑफ रेप्लिकेटिव सेनेसेंस. And the sixth is increased angiogenesis. Seventh is ability to invade and metastasize. And the eighth is evasion of the host immune response. तो ये सारे 8 पॉइंट्स थे. ठीक है, मैंने रामदास नाइक से लिए रेफरेंस. तो ये इसमें हम पहला पॉइंट जो है इंक्रीज्ड एक्शन ऑफ पॉजिटिव ग्रोथ रेगुलेटर्स को अच्छे से समझने की कोशिश करता हूँ. करेंगे हम सब साथ में. ठीक है, लेट्स सी. यहाँ स्क्रीन पे आप लोग देख रहे होंगे नॉर्मल सेल कैसा होता है. हाउ द नॉर्मल सेल लुक्स लाइक. So you can see everything is perfect, right? Nucleus is in shape, nucleus you can appreciate, vacuole is there, mitochondria is there, Golgi body, ribosomes, everything is there, right? Nicely. Now let's see. ये आप देख सकते हैं, this is the normal cells and this is the cancerous cells, how they look, right? So if you'll see in the cancerous, the red one, it is the many cells that continue to grow and divide, right? And you are seeing there is a variations in the size and shape of the cells. काफ़ी differences हैं इन दोनों के साइज़ में. अगर ब्लू वाला और अगर आप रेड वाले कंपेयर करें तो ब्लू वाला का यू कैन अप्रिशिएट द साइज. आप बोल सकते हैं कि इट्स ओवल और राउंड काइंड ऑफ. नाउ यू कैन सी इफ यू अप्रिशिएट द न्यूक्लियस कंपेयर नॉर्मल सेल्स में. इन द नॉर्मल सेल्स दे आर ओके नॉट दैट मच लार्जर इन साइज, बल्कि कैंसर सेल में यू कैन सी द न्यूक्लियस, राइट, इट इज़ लार्जर एंड सो डार्कर देन द नॉर्मल. और आप देखेंगे देयर आर एबनॉर्मल नंबर ऑफ क्रोमोसोम्स, राइट, एंड जो कि डिसऑर्गेनाइज्ड फैशन में अरेंज है कंपेयर टू द नॉर्मल वंस, राइट. एंड यू कैन सी देयर इज अ क्लस्टर ऑफ सेल्स विदाउट अ बाउंड्री, राइट. डिफ्यूज पैटर्न में अरेंज है, यहाँ पे देख सकते हैं एक तरह से. और यहाँ पे ये प्रॉपर बाउंड्री यू कैन अप्रिशिएट, हम देख सकते हैं कि बाउंड्रीज हैं. ठीक है. एक और डायग्राम मैंने रखा है यहाँ पे कि नॉर्मल और कैंसर सेल में यू कैन सी नॉर्मल सेल में लार्ज साइटोप्लाज्म, सिंगल न्यूक्लियस, फाइन क्रोमेटिन, राइट. whereas in cancer cells you can see small cytoplasm, multiple nuclei, and there is a large nuclei, it's multiple nuclei and coarse chromatin. ये differences generally जो हम observe करते हैं, what we can observe, वो हम यहाँ देख रहे हैं. Our the first topic is cell proliferation. Not cell proliferation, that is the hallmarks of cancer, that is self-sufficiency in growth signals, right. We are discussing that things. Before that I would like to draw the attention that it's written in the book that normal cells may undergo malignant transformation by corrupting the normal steps of the cell proliferation. The normal steps, right. If that any of the normal steps of the cell proliferation is getting corrupted, means what? Getting disrupted means there is some kind of interference in that cell proliferation steps. Because of that, it is leading to a malignant transformation, right. What happens that to proliferate, right, if a cell wants to proliferate, अगर एक सेल proliferate करना चाहता है, it needs growth signals. okay, growth signals की ज़रूरत है. क्या होता है, जो हम पहला पॉइंट डिस्कस कर रहे हैं, that we are discussing the first point is self sufficiency in growth signals. The cell that has gone under the malignant transformation, यानी कि वो ट्यूमर सेल बन चुका है, वो किसी पर निर्भर नहीं है. इट्स नॉट डिपेंडेंट ऑन एनी अदर growth signals. That growth signals will come from somewhere and then it will get activated. Here the cell is self-sufficient in producing the growth signals, the growth factors. He can make that growth factors. वो खुद से बना सकता है, उसे ज़रूरत नहीं है कहीं से लेने की, right? Now, so it's just written here, the growth signals, that is the cell is self-sufficient itself and it can produce its own growth factors. There is no need of the external source, right? Now, second thing that you need to understand is the gene is normal, right? When the gene is normal, the other name we can give it as proto-oncogenes. नॉर्मल जीन्स को हम प्रोटो-ऑन्कोजीन्स कहते हैं. अब ये जीन्स जो कि नॉर्मल स्टेट में है तो ऑब्वियसली प्रोटीन्स प्रोड्यूस करेगा. दे विल प्रोड्यूस द प्रोटीन्स एंड दिस प्रोटीन्स विल रेगुलेट द सेल प्रोलिफरेशन, ऑल्सो द डिफरेंशिएशन एंड द सर्वाइवल ऑफ द सेल. ये सारा प्रोटीन्स का काम है जिसमें वो हेल्प करेगा, राइट? नाउ तो जस्ट अभी हमने देखा कि जो नॉर्मल जीन है उसको हम प्रोटो-ऑन्कोजीन बोल रहे हैं, राइट? वी आर टेलिंग इट ए प्रोटो-ऑन्कोजीन. Now, if there is a mutation in the proto-oncogene, means it is converted to oncogene. ठीक है? जब ये oncogene यानी कि अब म्यूटेशन हो चुका है, now it's an oncogene, so this oncogene will produce definitely that is the oncoproteins, right? Oncoproteins produce करेगा. Okay? Now, this oncoproteins will further contribute in the cell proliferation steps. जब ये ऑन्कोजीन्स बन जाता है, when the it is now in oncogene, so it promotes its autonomous cell growth, like it's not dependent on anyone. Okay, and when a proto-oncogene is converted to oncogene, so no one can control its activity, right? So its activity cannot be controlled. Okay, now so this is the diagram which I have drawn. This picture, this is basically a cancer cell. What has happened inside a cancer cell? That there is no control over the activity of the cancer cell. Okay. Now, how does normal cell responds to growth factor? To understand the abnormality, we should know what is the normal first. What is normal happening there? Okay, because the interfering the normal process causes the abnormalities. Okay, so let's dive into this more detail. So here in the slide you can see I have the peak on the left side. So this is the peak is telling how a cell cycle progression happens, how a cell proliferation happens, how a cell is going to proliferate. And on my right side it's written where the interference can happen, where the corruption can happen. Okay, where it can get corrupted. कहाँ पे गड़बड़ी हो सकती है, किस स्टेज में गड़बड़ी हो सकती है, वो हम लिखा हुआ है. तो गड़बड़ी ग्रोथ फैक्टर के पॉइंट पे भी हो सकती है. इट कैन हैपन एट द पॉइंट ऑफ ग्रोथ फैक्टर, लाइक एक्सेस प्रोडक्शन ऑफ द ग्रोथ फैक्टर. इट कैन हैपन एट द ग्रोथ फैक्टर रिसेप्टर. अगर ग्रोथ फैक्टर रिसेप्टर ज़्यादा प्रोड्यूस हो रहा है, इट्स प्रोड्यूस्ड इन अ मोर क्वांटिटी, we'll see in detail later, then also it can lead to the what formation of the cancer cell. Agar there is a protein involved in the signal transduction, in that protein also is some kind of overproduction or mutation in their gene producing this kind of proteins, then also it can lead to the production of the cancer cells, formation of the cancer cells. Like that, in the nuclear regulatory protein, the aberration may occur. The aberration may occur in the proteins that are regulating the cell cycle. Very important. So these are the 5 right points which we will deal in the coming video, right? So एक कैसे सेल प्रोलिफरेशन हो रहा है, सेल साइकिल कैसे प्रोग्रेशन होता है, उसको समझने के लिए मैंने एक डायग्राम बनाया है, फ्लो चार्ट. Okay, that diagram which we saw in the previous slide that I have drawn, okay, here to make you understand that what you are seeing in the screen right now, so this yellow thing is a bilipid layer, right? This is a cell membrane. Okay, in that this red color you are seeing is, this is growth factor receptor, means where the growth factor will come, this blue color, sky blue color thing will come and bind to this red color, that is the growth factor receptor. Now this has come and bind here. Okay, and this is a site for the bind of the growth factor in the growth factor receptor. जब ये receptor के bind करता है, then what happens that this red thing, they are nearby, right, situated nearby, so they are दोनों आस पास है, right, so they got dimerized. Dimerized means they got paired up. अब इनके पेयर होने से, now they have become paired, this will lead to the activation of some kind of binding protein that is situated in the cytoplasmic side of the cell membrane, right? ये cytoplasmic side है, यहाँ पे ये present है, and इससे associated है, there is a RAS protein, there is a RAS protein, and this RAS protein is getting activated. Now this RAS protein is getting activated, and when this RAS protein is activated, this will further activate the signaling pathways. There are two signaling pathways, आप देख सकते हैं यहाँ पे, RAF and this PI3K pathway is activated. Now this PI3K will further activate the AKT pathway, and this AKT will further activate the mTOR, and this will lead to the activation of the transcription. Similarly, this RAF will further activate the MAP kinase, and this MAP kinase will further stimulate or activate the process of transcription, and this transcription will lead to the by the MYC protein, and this MYC protein will lead to the cell cycle progression. This is just general overview जो मैंने बताया आपको, which I told you. Okay, that कैसे, how, when there is a growth factor coming in a normal cell, how the process is happening, how it is leading to the cell proliferation. There is a binding of the growth factor to the growth factor receptor. This growth factor receptor dimerizes, leading to the activation of the RAS. RAS protein activated causes the activation of the further two pathways, which helps in the activation of the transcription process led by the MYC protein. This MYC protein has a very important role, and this correlates to the causes further cell cycle progression. Okay, one more thing, once this RAS is getting activated, so this activated RAS can come back to the normal state, also this inactivated state. It happens by the GTP hydrolysis, or hydrolysis of the GTP. So here you can see, this is the RAS, right, activated one, and this is the RAS inactivated one. So there is a GTPase enzyme that is causing the hydrolysis of GTP, which is breaking a GTP into GDP and inorganic phosphate, right? So we will see this further, that what is RAS, what is there in RAS that is it getting activated, what's happening there. If you will see that RAS is bind to the GTP, then it is activated. Once when the RAS is bind with the GDP, then it is inactivated. So this is the basic concept behind this. Okay, so now don't get confused with the side written things, just focus on this pathway. What happens, just we discussed that growth factor binds to growth factor receptor, this gets dimerized, means pair up, and this leads to the binding protein attached to the growth factor receptor that goes and binds with the RAS protein, right? And this RAS protein gets activated, that is called RAS activation, and this further causes the activation of the two pathways that we already discussed. What are those two? The pathways are RAF ERK MAP pathway, right, and the other one is PI3K AKT pathway, right. इससे क्या होता है, this leads to the activation of the transcription, and transcription causes the formation of this called here MYC protein है, and this and this MYC protein causes the cell cycle progression, right? So this is the normal thing what we discuss, right? So what happening, literally what happening there, that is causing this kind of abnormalities, like leading to the cancer cell formation. Okay, so there is something wrong in this pathway, कुछ गड़बड़ी है इस पाथवे में, कहीं गड़बड़ हो जा रही है. तो उसको हम डिटेल में देखते हैं, राइट. तो पहला गड़बड़ी, जो फर्स्ट error that is happening is there, the first aberration is happening is there in the excess production of the growth factor. What is there? Excess production of the growth factor. तो growth factor का excess production हो जा रहा है, right? Now suppose one cell is causing excess production of the, like they are forming growth factor by their own, right? Obviously that will act on them also, and that will act on the cells nearby to them, right? So that's why you can see the paracrine action and the autocrine action. Okay, so you can see here, right, there is a paracrine action on this cancer cell, and you can see this cancer cell is now itself, it's producing, right, excess of growth hormone. It's acting on its same receptor, that's insulin growth like factor 1 receptor, right? So here you can see that here the paracrine, right, this cell, this stromal cell is producing excess of the growth factors, and that is coming and binding to the receptor of the cancer cell. But here you can see that this cancer cell itself on the right side itself is producing insulin like growth factor 1 excess, right, and that is binding to its own receptor, that's the growth factor receptor, right? This is called autocrine action, and this is called the paracrine action. Okay, the left one is the paracrine action, the right one is the autocrine action, right? Just one example I have added, don't confuse with the example, just if you can, just you can remember that in the glioblastoma, okay, this now is a tumor cells, right, they produce excess of the platelet derived growth factor, and also they remember they excessively produce this platelet derived growth factor receptor also. This much only, just remember. If you can remember this example, otherwise just understand what's happening there. Okay, so first topic is covered, that's the growth factor. What happens at this stage is can be excessively produced, excess production होगा इनका, और ये paracrine and autocrine action होगा, right? Paracrine autocrine action we saw, right? Next, coming to the this second part, जहाँ पे growth factor receptor dimerize होते हैं. Dimerizes, right, after the binding with the growth factor. So it needs someone, right, to dimerize. It needs the growth factor. इनको जरूरत है डायमराइज. अगर ये करेंगे आपस में, like we saw here in the diagram, we saw like they are dimerizing, so they need growth factor, then only they will dimerize. But ऐसा होता है, like it will happen like that. Now they don't need any growth factors, they will be constantly in this dimerized state itself. They don't need any growth factor to get dimerized now. Means they will constantly be in on phase. हमेशा वो एक्टिव रहेंगे, हमेशा इस डायमराइज फेज में रहेंगे, और ये हमेशा ऑन रहेंगे, राइट? सो इनका जो ये ऑन रहना है, लाइक कॉन्स्टेंटली डायमराइज स्टेट इज नॉट ए नॉर्मल थिंग, राइट? In the absence of growth factor, if they are dimerized, that's not a normal, that's an abnormal thing, right? So that's what I have written here. The receptor is always in paired state, that's the aberration here, right? Even no growth factor is bound to it. Okay, so here you have to understand that how this mechanism is happening. So this mechanism is happening by the gene amplification can happen, right? And whether there can be a mutation in the gene, right? Now इसको समझने के लिए, to understand this is more detail, if you want I can tell you, so you can skip this part also if you know already this, right? But I will like to add the gene amplification. What's happening in the really here in the gene amplification is, see there is gene amplification means like gene है तो उसका hundreds of copies बन गया, right? hundreds of copies बन गया, बहुत सारा copies बन गया सेम जीन का, जिसकी वजह से क्या हुआ, वो जीन में किसी भी प्रकार का म्यूटेशन नहीं हुआ, बस एक नॉर्मल जीन था, उस तरह का 100 जीन बन गया जो कि नॉर्मली है, राइट? दे आर नॉर्मल ओनली, सो दे विल बी कॉल्ड इट्स proto-oncogene only, they are not became oncogenes, right? They have not converted into oncogene. They are now proto-oncogene only. But the normal gene only is the more in number, right? So obviously sir, proto-oncogenes means they will be forming normal proteins. So उससे क्या हुआ, like this normal proteins is now more, right? So overproduction हो गया normal proteins का. So because of this, right, the stimulation is more they are getting stimulated, the pathway is getting more stimulated, and that is causing what, the further proliferation of the cell. अंडरस्टूड. सो इसका मतलब होता है, यही है जीन एम्प्लीफिकेशन का मतलब. नाउ जीन एम्प्लीफिकेशन कैन हैपन फर्दर, राइट, बाय टू वेज, बाय HSR एंड बाय एन्यूप्लोडिंग. ओके, तो व्हाट इज HSR and it's mean homogeneous stainable regions. So homogeneous stainable regions is basically क्या हुआ कि एक क्रोमोसोम्स थे, राइट. सो यू कैन सी इन दिस डायग्राम, जैसे कि यहाँ पे क्या हुआ कि MYC जीन्स थे, राइट. इस रीजन में क्या थे, MYC जीन्स थे, यस और नो. सो अब क्या हुआ कि यहाँ पे जो MYC जीन की जो रीजन थी, ये वाली, यहाँ पे बहुत सारे जो जीन्स की जो कॉपीज़ थी, जैसे मैंने बोला कि एम्प्लीफिकेशन हो गया, तो अगर एक जीन थी तो यहाँ पे बहुत सारे मल्टीपल कॉपीज़ आ गए. हाँ है ना, बात समझ में आई. तो ये जो बहुत सारे मल्टीपल कॉपीज़ आ गए, एक तो ये हो गया, तो इनको आप बोल सकते हैं डबल मिन्यूट्स. इसको आप शॉर्ट फॉर्म में डी मीन्स भी बोल सकते हैं, क्योंकि ये जीन के वो 100 कॉपीज़ बन गए सपोज यहाँ पे, क्योंकि एक ही जीन रहना चाहिए, तो इसके इस क्रोमोसोम पे आपको क्या मिल गए, 100 ऑफ कॉपीज़ बन के रेडी हो गए, राइट. नाउ अब मैंने बोला कि HSR क्या है, होमोजीनस स्टेनिंग रीजन. HSR में क्या होगा, व्हाट हैपेंस हियर इज दैट, राइट, दैट देयर इज ए अल्टरेशन इन द क्रोमोसोम, दैट क्रोमोसोम इज नाउ अल्टर्ड, राइट, बिकॉज ये यहाँ पे ऑलरेडी जीन्स इस जगह पे लोकेटेड थे. बिकॉज ऑफ इट्स मल्टीपल ऑफ कॉपीज, now this gene you can see is has, due to the increased copies of gene, right, this has occupied the new location in the chromosome, right. तो ये लोकेशन इस जगह पे थे, राइट, टर्मिनल एंड पे थे, राइट. सो यहाँ पे यू कैन सी ये सेंट्रोमियर वाले पार्ट पे यू कैन सी देम एज द पार्ट ऑफ द सेंट्रोमियर तक ये एक्सपैंड कर गए हैं, राइट. सो दैट्स वाई, so that's obviously away from the normal location of the involved genes, yes or no, and they appear as a homogeneous, right. In the G banded karyotype if you will do, in that they appear as a homogeneous, right, एक जैसे दिखते हैं. that's why we are telling as a homogeneous staining region. So these are the two patterns which you can see. First pattern you can see that, right, like this, that is the double minutes, because they are the extra chromosomal multiple small structures, right. And further this there can be a chromosome alterations that can be referred as a homogeneous staining region, right, because this increased copies of the genes here is inserted to the new chromosomal locations, right, that can be distant away from the normal, that is distant away, right, distant away from the normal location of the involved genes, yes or no, normal location of the involved genes. That's the thing, concept behind this, which you can see in the book, right, that I have already told you what is the homogeneous stainable regions. Next, and I told you about the aneuploidy. So aneuploidy means, so there is a, you can see the chromosomal, right, so that is not in the multiple of the 23, that is also you can see. So we understood. So here we were, that receptor is always in the paired state. This is the aberration, right. In the absence of growth factors, they are in paired state. And why this is happening? Maybe there will be mutation, right, or there may be gene amplification. Understood? So this was the aberration what we can see at the level of growth factor receptor. Okay. Now we are looking at the level of signal transducing oncoproteins. So what is happening here, that there is a RAS gene, right? Simply there is a RAS gene. Okay. This RAS gene, obviously what it gene produces proteins. The RAS gene will produce RAS proteins, yes or no? Now this RAS protein is in an inactive stage. If you zoom it, what you will see after zooming means you will see RAS is with GDP, right? Now to become activate, to get activated, what is happening, that the RAS is getting activated means you will see here the RAS is with GTP, right? RAS is with GTP, and this is a cycle. If you will see, this is a cycle happening here. What cycle you will see here, that this GTP is, that is bind with the RAS, is hydrolyzed by the enzyme GTPase, and this GTP is converted to GDP, and this RAS is only with GDP means this RAS is now in inactive stage. This is a simple mechanism which you can see here that is happening here, right? Now if you will see in our body, there is a RAS means there is a proto-oncogene, obviously means it's normal gene, right? Healthy gene. So three types of RAS you will see: KRAS, HRAS and NRAS. These are the RAS genes. And what happens where the aberration is happening? Let's talk about the aberration part. That is the RAS proto-oncogene is a normal gene. Due to the point mutation, you will convert to the RAS oncogene, right? Now this RAS oncogene, what's happening? क्या हो रहा है RAS oncogene में? See, there was a cycle happening, right? So if you will see this, I have rounded with the orange color. This cycle was happening, right? Between RAS activation and inactivation, this cycle was happening, yes or no? So this cycle is happening because this GTPase will hydrolyze this GTP which is bound to the RAS, right? So what happens after the conversion of the RAS gene to RAS, means RAS proto-oncogene to the RAS oncogene, due to the point mutation, this ability to get rid of the GTP is lost. Means this RAS and GTP is trapped now, right? This GTP can't escape this RAS, right? It cannot get hydrolyzed. Means the RAS plus GTP will always be there in the bound form. Okay, so that's what you can see here. The RAS plus GTP is trapped now. Okay, so they are trapped, right? Next, just a little extra information in this I would like to draw your attention towards, that there is a ABL gene in our body. Okay, now this ABL gene is basically not associated with any receptor like the RAS was there. Okay, it is a non-receptor associated tyrosine kinase. Okay, non-receptor associated tyrosine kinase. This is seen in a case of the CML. CML everyone will be knowing, or even you are not knowing, CML is, you will learn in further coming chapter. This is chronic myelogenous leukemia. Chronic myelogenous leukemia, right? So in this what happens is, there is a ABL gene on the chromosome number 9. This ABL gene on the chromosome number 9 goes and joins the BCR gene located on the chromosome number 22. Remember this, it joins on, means this ABL gene, right? This is ABL gene is going to the BCR gene that is located on chromosome number 22. Now what happens because of this? Tell me. You will see this BCR ABL thing, right? This BCR ABL thing is now called a fusion gene. Now this is a kind of new gene formed into the market, right? You can also call it as a chimeric gene, right? So because of this, the ABL gene was fine, right? That has become oncogene, right? That is converted to oncogene, that is cancerous, and it will further produce the what? Oncoprotein, and means danger, right? We don't want to discuss now in detail because this is some other topic we're discussing. Okay, next, this is, remember, it's in danger condition. Okay, now coming to the further stage, in this stage of the MYC protein thing, right? So all the signal transduction pathways, okay, this all the pathways which we either this is the RAF, ERK, MAPK pathway, whether this is PI3K-AKT pathway, this will come and this will activate or stimulate the nuclear transcription factor, and this nuclear transcription factor, right, will bind to the DNA and regulate the transcription of genes. Okay, and you know the transcription is a process that in which the RNA is synthesized from the DNA, right? And the transcription factors will stimulate the growth promoting genes which activate the cell cycle. So you can see here that there are many genes, right, like MYC, MYB, right, JUN, FOS, REL. Okay, so there can be a mutation in this kind of genes. We are mostly discussing the MYC gene because the MYC gene mutation is mostly involved in the human tumors. So what happens that there is a mutation in the MYC gene, right? And because of this, the MYC protein production has increased. Because of this, so MYC protein is basically a nuclear regulatory proteins. न्यूक्लियर रेगुलेटरी प्रोटीन सिंपली इसके नाम से आप कह सकते हैं कि दिस न्यूक्लियर रेगुलेटरी प्रोटीन्स will stimulate the growth promoting genes in the DNA, obviously. So means they are going and binding, this proteins goes and bind with the DNA thing, right? And then they will promote the further cell cycle. And who are those these growth promoting genes? They are basically cyclins, right? So ये process चलता है, this process happens. Now, so if obviously, so because of this, the cell proliferation will be more and more if the MYC protein production is more. Okay, and why this is production is more, it's happening because it's happening because of the mutation in the MYC gene, right? And you will see that there is a MYC, 3 MYC, you will see gene CMYC, NMYC and LMYC. Okay, you will see the 3 types of gene in our MYC gene in our body. Okay, these are CMYC, NMYC and LMYC. And I have not discussed what's happening because of the overproduction once the mutation is happening in all these 3 genes, because this C1 is, just remember, it is associated with the Burkitt lymphoma, and MYC related to the neuroblastoma because N means neuroblastoma, and L means, you can remember, lung, that is small carcinoma of lung. Okay, so C for Burkitt lymphoma, N means neuroblastoma, and L for a small cell carcinoma of the lung. You can remember like this. Okay, now we are reaching towards the end of the session. So this is the fifth stage, where all the growth promoting stimuli, you can see here, all the growth promoting stimuli, okay, they causes the entry of the cells which are in the quiescent stage into the cell cycle. Means they stimulate the proliferation, simple. So, but there are some cyclins and there are some CD kinases, cyclin dependent kinases in our cell that regulate this cell cycle, that regulate the cell cycle. The cyclins majorly you can see cyclin D, cyclin E, cyclin A and cyclin B. Now, there are cyclin and there are cyclin dependent kinases. They both combine and they form CDK cyclin complex. The CDK cyclin complex causes phosphorylation. What they causes? They causes the phosphorylation of the essential proteins, and because of this the cell cycle gets activated. Now obviously there should, this is normal thing what we are discussing, what happening normal as a normal cell, in a normal cell what's happening. Okay, now obviously their cell cycle is happening, there should be some negative control over the cell cycle. So that negative control is being made by the cyclin dependent kinase inhibitors. What they does? They do silencing of the cyclin dependent kinases so that excess proliferation should not happen. So anything which can come and interfere with this, anything which can come and causes the decrease in their number, okay, it means CDK silencing will be less, and if the CDK silencing is less, means there will be more formation of the CDK cyclin complex, and there will be more phosphorylation of the essential proteins, and there will be more and more activation of the cell cycle, right? So who is causing the decrease in the number of the CDKIs? Let's see. So who are they? They are basically the two pathways, these two pathways, RAF, ERK, MAPK, and this PI3K AKT pathways. Obviously that we discussed there, right? You are right. We are talking about these pathways and these pathways. Okay. So these two pathways are also called. So these two pathways are also called as a mitogenic signaling pathways. So this mitogenic signaling pathways is increasing more and more, right? Because of this, why this is increasing more and more? Because the further, not further, the previous, the four aberrations what we studied, they all were promoting this mitogenic signaling pathways, and if this will be more, then there will be a down regulation of the CDK inhibitors, means CDK inhibitors will become less, they are less produced, and because of this the cell cycle will increase more and more, because now the negative control is lost, which is being given by the CDK inhibitors now, because they are not in much number. Okay, so you can see cycling D genes overexpression also is a major factor, and we can see there is a CDK4 gene amplification in this case. So if there is a more and more cyclin D gene overexpression, the more cyclin products will be formed, they will, and if there is a CDK4 gene amplification, understood, so their productions will be more, and they will bind, they will the CDK, right, cyclin complex, this is a complex is formed, and they will cause a phosphorylation of the essential proteins, right, and will further cause the cell cycle activation. You should know. So it's a simple mechanism which we discussed. So let's summarize. This was a summary part thing is, we saw the cell proliferation steps, how they are getting corrupted, means they are getting interfered by some kind of mutation, right, or amplification of the gene, how difficulty is in the happening to the normal cell, how they are becoming a cancerous cell. Now growth signal was the major factor, was the first step, right, or the growth factor, you can say that the cell was depending on this growth factor for the proliferation, right. Now this growth signal the cell is able to prepare by themselves, right, so they are due to some, obviously some mutation, so they are not dependent on anyone, and that is in cancer cell. Now we understood the what is proto-oncogenes. Proto-oncogenes are nothing but they are the normal genes only. Okay, so what happens, the normal genes will encode some of the proteins, here example like nuclear proteins, and this nuclear proteins will regulate the cell proliferation, differentiation, and the survival of the cell, right. Now this proto-oncogenes, that is a normal gene, right, this is a normal gene, is getting mutated and forming oncogenes. Oncogenes form further oncoproteins, right, and now this oncogene, this is a stage that is happening autonomous cell growth, right, that the activity cannot be controlled by anyone, so this is nothing but a cancerous cell. Okay, we discussed how the normal cell responds to the growth factor, and how at the all stages there can be a chance of aberration, right? How the aberration can happen, and these are the 5 points where the aberration can happen. We discussed that also in detail. This was a diagram which is very important. You have to practice this, right? This was the main one page flowchart for all those 5 places where aberration can occur, right? And we learned the HSR part also, what is homogeneous staining region and all. So thank you for watching video. Any queries you can comment down, okay, and share with your friends, like it and subscribe it. It really gives a motivation to prepare the next video. Thank you. Signing off.